Before watching this video, please read or review Chapter 2, Neuronal Physiology. What is electrophysiology? Electrophysiology, also known as neurophysiology, is the study of the electrical activity of a functioning biological system. We can use electrophysiology to measure the electrical activity of specific cells, usually neurons, to understand how they function within the nervous system and how their activity may be related to behavioral and cognitive processes. There are a number of different ways to gather electrophysiology data. A number of these methods involve inserting an electrode or electricity measuring device into tissue or cells. Most modern electrophysiology rigs are capable of both measuring and injecting current, as well as measuring and adjusting the voltage or membrane potential of cells through a single electrode. This recording electrode is paired with a reference electrode that sits in the bath next to the tissue or cells to provide baseline information. Here is a real electrophysiology setup for recording cells in a live brain section. You can see the reference electrode immersed in the solution and the recording electrode not yet inserted into the tissue. Also shown are the ACSF perfusion and vacuum lines that continuously perfuse the slice in fresh, artificial cerebrospinal fluid. On the right is the thermistor, which measures the temperature of the solution and connects to a piece of equipment off-screen to adjust it. Next, let's talk about types of electrophysiology recordings. There are two main types of electrophysiology recordings, extracellular recordings and intracellular recordings. Both are extremely valuable and both have pros and cons. Extracellular electrophysiology recordings are taken when an electrode is inserted into a brain area or next to a number of cells. Using this technique, we can gather electrical data from a lot of cells at once and develop a sense of how a region or multiple cells, such as the hippocampus, may function collectively. However, because we cannot access the inside of the cells, we cannot collect as much data from each individual cell. In contrast, when we collect intracellular electrophysiology recordings, we gather data from a single cell at a time. This is more time-consuming, but it gives us far more precise information about the electrical activity of one individual cell. Conducting multiple experiments using both of these techniques can help us understand how groups of neurons function and how individual neurons within those groups function. To collect electrophysiological data from cells or animal models, researchers make use of three different types of tissue preparations. Individual neurons or other electrically active cells can be grown or cultured in vitro, meaning outside of an animal. These individual cells can be targeted for intracellular electrophysiology recordings. Slices of fresh brain, whole brains of smaller animals, or isolated spinal cords, nerves, or nervous systems can be used for electrophysiology. This tissue can be used for both intracellular recordings from individual neurons and extracellular recordings from groups of neurons that communicate with each other. Electrodes can be inserted into a living, anesthetized animal. After recovery from surgery, researchers can collect intracellular recordings from individual cells or extracellular recordings from groups of cells in a living animal. These recordings can be taken as the animal engages in behaviors such as navigating a maze or making a decision. A quick side note, EEG, or electroencephalogram recordings, are another example of in vivo electrophysiology. EEG is primarily used in humans and involves applying electrodes to the scalp to measure the electrical activity of the brain. It is covered in depth in another section of this book. So now that we know a bit about how it works, let's talk about what electrophysiology is used for. Intracellular electrophysiology can be used alongside pharmacological tools to understand how ion channels support neural signaling. On the top is an open sodium channel, which allows sodium to flow into the cell. We know that these sodium channels are necessary for neurons to generate an action potential, but how do we know that? Researchers applied a sodium channel blocker, known as tetrodotoxin, or TTX, to neurons. When recording neuronal activity, they found that neurons with blocked sodium channels did not generate action potentials. Thus, they discovered that sodium channels are critical for action potential generation. Studies like these have advanced our understanding of the mechanisms of neural signaling and improved our understanding of diseases that involve impairments in neural signaling, such as epilepsy and schizophrenia. Electrophysiology can also be used to measure the activity of cells within a neural circuit, or group of cells, that fire together to support specific functions. We start with a group of neurons shown as triangles. Some of these neurons receive input from a different brain region. These neurons are shown in blue. But how do we actually know which neurons receive input from this other brain region? One way to discover this is electrophysiology recordings. We can stimulate afferents from the other brain region and record the response of individual neurons. In this example, we are recording from neuron 1. When we stimulate the afferents, neuron 1 fires an action potential. This tells us that neuron 1 receives direct input from the other brain region. We can repeat the stimulation, but record from another neuron, neuron 2. When we stimulate the brain region afferents, neuron 2 does not fire an action potential. This means that neuron 2 does not receive direct input from the other brain region. We can repeat these recordings many times to discover how specific types of neurons may preferentially connect with other brain regions. This helps us understand the flow of information between brain regions and between neurons within brain regions. But please remember, this is not the only way to gather this type of information. Neural circuits also exist between neurons within the same brain region. One way to analyze these neural circuits is to record from two neighboring neurons at the same time. Then we can force neuron 1 to fire action potentials, in blue, and record the response in neuron 2, in purple. If neuron 2 responds, it means that neuron 1 synapses onto it and these two neurons are connected. Using this technique, we can gain a lot of information about how strongly these neurons are connected, which is important for understanding how neural circuits form and function. In vivo, an electrode placed in the visual cortex of a living mouse can reveal which neurons fire together when the mouse sees a specific stimulus. In this example, the mouse is looking at vertical bars. Prior studies used in vivo electrophysiology to discover that specific neurons in the mouse's visual cortex, the green and yellow neurons in this figure, respond only when bars are in specific orientations. These same neurons may not fire action potentials if the bars were horizontal instead of vertical. These electrophysiological data can also be collected in animal models of diseases to see how specific diseases affect the wiring of neural circuits. To conclude this video, I'll walk you through an example of intracellular electrophysiology recording from brain tissue slices. I will use an intracellular recording technique called whole cell patch clamp electrophysiology, or patching. To patch a cell, I break through its membrane without killing it. This gives me the ability to both record from and control the electrical activity of one neuron at a time. On the top left of the screen, I am showing a drawing of what happens in the pipette and the cell. On the bottom left is a recording of my movements to patch a cell. On the top right is a recording of the resistance of the pipette tip. I use this resistance to tell me when I am up against the cell membrane and when I have broken through or patched the cell. On the bottom right is my tissue and the pipette I will use to patch, as seen through a microscope. For the first part of the video, pay close attention to the bottom right and see if you can spot the pipette I am using to patch and the neuron I am aiming for. A quick hint, both are slightly to the left of the middle of the screen and will go in and out of focus as I move the microscope and pipette around. I identify a neuron that looks healthy enough to patch. Here, I am looking for neurons in the neocortex, specifically in layer 5 of the primary visual cortex, and I've identified a neuron here. Then, I bring my recording electrode into focus under the microscope camera. I next lower the electrode until I am near the cell I want to patch. I know I am about to touch the cell I want to patch because the pipette resistance in the top right image goes up by 0.1 megaohms. Once I touch the cell membrane, I pull it into the electrode tip until it is sealed. I know that the cell membrane is sealed against the pipette tip because the pipette resistance in the top right image is now over 1 gigaohm, indicating a tight seal. Then I very carefully and gently pull on the membrane until it breaks. As soon as it breaks, I release the suction so that I don't suck the inside of the cell into the electrode. I will know that the membrane has broken because the resistance will drop rapidly. Rewatch the past few seconds of this video a couple of times. See if you can spot the resistance change that tells me I've broken through the membrane. I've patched a cell. Now I can both record and control its electrical activity. I can record the resting membrane potential, shown here, Or I can force it to fire action potentials by injecting positive current through the electrode, shown here. Here, I first inject current at subthreshold levels which transiently depolarize the cell, but not quite enough to reach an action potential. You can see as the membrane potential of the cell reaches minus 40 millivolts, it has depolarized enough to generate an action potential. All current injections after that one are enough to evoke more action potentials. In conclusion, electrophysiology is a vital tool for neuroscience. We can use it to both record and control the electrical activity of neurons or other cell types. Thus, electrophysiology is incredibly useful for understanding how individual neurons or groups of neurons in a neural circuit function in both healthy and diseased states. This video has been a very brief introduction to some key electrophysiological techniques, but please keep in mind there are many more techniques I haven't mentioned that are also incredibly important for understanding how neurons function and how they connect with each other.